Welcome to Duchenne Science Live, brought to you by Action Duchenne. My name's Neil.
And I'm Sid.
And this week we're going to be looking at a paper that looks at metabolism in the muscles of people living with Duchenne and how those processes in the cells might have changed.
So metabolism is all the thousands of chemical reactions happening in a cell, all your cells, of all your tissues, of your entire body, all of the time to keep you going.
Yeah, so I think the kind of simple definition of metabolism would be the kind of taking in food, burning the food to get energy to do something, right? It's those processes, it's those reactions.
So essentially, fuel going into the body, release of energy used to do stuff, whatever that might be, break things down, build things up, and removal of waste products. And so that's the sort of basic linear step by step. But for each of those steps, for each of those processes, you've got thousands and thousands and thousands of chemical reactions going on.
Yeah.
And those reactions need to be done in a controlled way. And so this wonderful, exciting diagram can pop up of like all the amazing things that are going on.
Yeah, because actually it's not a whole series of pathways, it's actually this whole network, isn't it? And they all interact. Yeah, so this diagram isn't really important. What's important is the complexity that the diagram shows and how much things interact and kind of fit with each other.
Yeah, so complex, they rely on one another.
So one of the big questions we should probably talk about is why we're talking about metabolism and Duchenne, because it's not necessarily an obvious link, right? It's Duchenne is a muscle condition. Why would we worry about the reactions?
Yeah, well, in the muscle tissues of the body, all of these reactions are going on. There's metabolism occurring in the cells of the muscles. So when a big important protein like dystrophin is absent or not the full molecule, it not only has a structural role in muscle cells, forming these bridges, these complexes, but it also has a role in regulation and signaling, which is it's all to do with all these pathways and all these reactions going on. So the absence of dystrophin or functional dystrophin is going to have a massive effect on the environment of the cell and all these reactions going on. So I was thinking about knock-on effects and the interlinking of things, and I thought about describing the cell as an ecosystem, as you would. So that's something in ecology in the natural world, and you're thinking how all the plants and the animals are interlinked, and so if you do something to one, it affects many of the others. And there's a lovely example of this, a story from Yellowstone National Park in the US. They introduced 14 wolves into Yellowstone National Park to control the deer. The deer population was massive. They were grazing and destroying all the young trees. The young trees weren't growing up. So it was a massive problem. And rather than cull the deer, they introduced 14 wolves. The wolves did hunt the deer, and they also drove the deer into certain parts of the park where the deer could hide from the wolves, essentially. So where the deer are no longer grazing, the trees are starting to grow up. And after a time, got a lot of regeneration on different plant species coming in. So we get different kinds of insects and birds coming back in. The trees are growing up. So now the beaver are coming back because they've got the wood to build their dams. So they're building their dams. They're starting to change the nature of the riverbank. The riverbank is also there's less erosion happening because the plants have grown up and they're stabilizing the soil around the riverbank. So actually, they found that there was a massive effect on the way the rivers ran in Yellowstone. They changed how they were running, and they had to redraw the maps. But once they stabilized and they were much more predictable, they would follow the same course season after season after season. So you might not have thought that introducing wolves to control the deer was going to have a major effect on the rivers of Yellowstone Park, but that's what happened via the series of steps.
I can't imagine people predicted that when they.
No, perhaps not. But it's in introducing the wolves and reducing the deer grazing, they have changed the environment. They've allowed regeneration, and it's having these amazing knock-on effects in the ecosystem. It just shows you how interconnected things are. So you can have maybe unexpected effects.
And actually, probably the complexity, the cells are incredibly complex. It's probably not a silly idea to compare a whole ecosystem in Yellowstone with, a muscle or a cell, because there's lots and lots of reactions going on constantly in cells, and they're constantly adapting and changing.
Yes, and there's up-regulating, so making and down-regulating, and this all has to keep the environment ticking over and feeding back and keeping things within parameters, so everything is functioning well.
In trying to put these networks together, it's probably important to understand this kind of thing, because there are drugs out there that might target some of these pathways. It might be possible to target them with new drugs, and people have said before that to treat Duchenne you might need more than one therapy for an individual. So maybe this is another target that somebody could investigate in the future. Still early days. I think it's important that we say that right up front. This is a first stage. They've developed a cool new technique that lets them look at all the metabolites in a muscle or in a sample. And in this paper, they're using that to see if there's any changes in the muscles of people living with Duchenne.
Yes, that's right. So it's a fancy new version of mass spectrometry to look at, as you say, levels of these metabolites. The metabolites are the various particular markers of biochemicals to show you what kind of reactions are going on at what kind of rates in these cells. And they seem to identify at least 37 key metabolites that were at levels either higher or lower than you'd see in the control samples of muscle in people without Duchenne. And they particularly said that there seems to be energy metabolism to do with fueling the cells and all the reactions that are going on inside them, and in phospholipid. So that's structural. That's about building things inside the cell and in building new cells. So essentially, that's all. If those things are out of whack, then you're going to see some big negative effects going on. And we looked through the paper and had and that looked great, and it looked like a way of identifying targets.
Yeah, and so 34, that's lots. That's plenty to go ahead and look at in the future and try and work out what you can target.
Yeah.
Try and work out the knock-on effects of targeting each one, because that's that whole network thing, isn't it? You target one of them and you're going to change the network, and so you've got to try and work out what would happen if you did decide to target one of them. And and so actually it looked really, really interesting, didn't it? And then we noticed a paragraph in the discussion of the paper, and I think this paragraph probably goes a long way towards explaining why we're doing these shows, right? That you can't just take the headline of a paper and say, oh, there's changes in muscle metabolites. And there's a list of the changes, so I know what I should be doing. You've actually got to read the whole paper because of paragraphs like this one. So the way they did this kind of study was to take stored muscle biopsies from people living with Duchenne. They've got nine people, and the muscle biopsies were before treatment was started, so they're before people started taking steroids. And so the idea is those muscle biopsies are, they've not been influenced by treatments. So you'd hope that what you're seeing in those is just kind of the muscle changes that you see in Duchenne. So that's great. And what they've done is follow that. Those samples have been in a biobank for years. So they've managed to follow those people in the clinic for several years so that they know what they look like in the clinic. They know what symptoms they're experiencing. And so they can say that actually these 9 people, they represent kind of Duchenne patients as a whole, I think is probably a fair. There's a selection of people who seem to be progressing slightly more quickly, slightly more slowly, and experiencing different symptoms, because we know Duchenne's variable, and that's what we see. So that's great.
Yeah.
And then the slight worry comes that in any science experiment, you use things called controls. And there's negative controls and there's positive controls. And the negative ones are there to tell you what it would look like if something didn't happen. Positives are there to show you what it would look like if something did happen. But then there's a whole other range of controls, and they're the controls that make sure you're really interpreting the result in the way in the right way, I guess.
Yes.
And so in clinical trials, you use a placebo. We talked about that last week. And that tells you that if you see an effect, it's because your drug is doing something compared to a placebo. In an experiment like this, you have to use some muscle samples from people who don't have Duchenne, because you're looking for changes that's caused by Duchenne. So you've got to have muscle biopsies from people who don't have Duchenne, muscle biopsies from people living with Duchenne, and you look for the differences. And that's what they've done here.
Yes.
And they found those 34 differences.
Yes. So in our lovely comparative study, and then we discover in the methodology toward the end of the paper that the muscle tissue samples that they use for their control, so what they're comparing the muscle metabolites in with people with Duchenne, they're comparing them to the muscle metabolites of people who don't have Duchenne, but they are patients. They have come in to a clinic because they have muscle pain and other symptoms, and the likelihood is that they have a form of fibromyalgia, so that's pain in the muscles of the body. So these people are not necessarily they're very likely to have metabolic differences in their muscle cells because they are coming in with a complaint or with a condition that is affecting their muscles and giving them pain and symptoms. So we're not looking at entirely healthy muscle tissue with a healthy metabolism. We're looking at muscle tissue that there's a problem with, and you'd expect there to be metabolic changes associated with that fibromyalgia condition. So can this, by comparing these two, can we really say what the profile changes for Duchenne are? Because we're not comparing them with another condition. It's really difficult to say what is true of Duchenne alone if you're comparing it with something else is going on.
I mean, it's important to say we understand why they've done it, because actually getting muscle biopsies from in a perfect world, you'd have muscle biopsies from healthy people of about the same age as your kind of population of interest, the people living with Duchenne. They mentioned in the paper that they could have got muscle biopsies from pediatric cases where people are having bone operations. And our first comment when we read that was, why didn't they use both? Because then it's more information. It's more useful information.
At least the age difference is no longer an issue, and it's not having a baseline. We talked about baselines in a different way last time, but if you're you want to know how metabolism is being effective in one.
You're looking for a change, aren't you? So
looking
it's important to know what it's changing.
Yeah, so if you're comparing it with something else, it could be that for those adult patients with who've come in to be diagnosed, then their metabolic changes are going to be could be considerable, and could be you can't say that it's all down. The differences aren't all down to one having Duchenne and one not, the other not. So.
But all that said, actually, I mean, the technique they're using, they've got this new technique that lets them look at lots and lots of metabolites really easily, really quickly, all at the same time.
Yes.
Actually, it's a fantastic technique. I mean,
yes. And that's great. And it could be honed and the problem of the control addressed somehow. Maybe just by, as you say, having
lots and lots of different controls,
different samples from different things going on, so you can see what is unique to one condition and not.
And actually, for families, Duchenne families, seeing this kind of research happening, it's great to see it happening. Just that people are starting to look at all the other aspects of the condition, because you have to understand all those knock-on effects that we were talking about. You've got to understand, if you could build a map of every change inside the muscle, that's the kind of information you need to really start designing or choosing targets that might help support kind of dystrophin therapies most, or might help you know, alleviate some of the symptoms. So I think it's really exciting that this kind of research is starting to happen.
Yes.
And
so for absolutely points for addressing exactly that sort of you know, problem solving and addressing the need for understanding by using the best and newest technology available. And it's great also because this kind of cool mass spectrometry can be used to assay things in less invasive types of samples. So it might be that if you get a good picture of the metabolites that appear in urine, it's far, you know, having collecting urine samples is going to be just so much easier than having to rely on muscle biopsies. But if you can have a picture of how metabolites change in different, or even a blood test, you know, as compared to a tissue biopsy, which is obviously a big deal.
Because actually, if you can understand everything that's happening in the muscles and then extrapolate to using a blood test or blood sample, that's
yes, but it is, and it's all helping in this tandem approach of dystrophin structurally and dystrophin in regulating the cellular environment, the ecosystem. And by addressing those both together therapeutically, you're going to have the best chance of having really much better outcomes.
Yeah, more effective treatments overall.
So it's great that this kind of research is happening and that they've tried to use this tool in this way. Maybe it's not quite, so we've pulled apart their control, but and so we wouldn't want to make any you know, statements based on what they've discovered, because we don't know what's down to the Duchenne exactly, but but in principle it's great, and it's a good approach to answering some questions.
A different paper and different type of paper to last week, and I think through these shows we're keen to explore as many different topics and as many different areas as we can for families, help to explain what's happening in the world of research. So if anyone's got ideas about what they want to hear, put them in the comments below. We'll try and pick up the papers, we'll try and do them in a future show. And if you've got any comments on this research, put them in the comments, and we'll try and answer any questions that you put there as well.
Give it a whirl. But yes, they're just encouraging everybody to not, not take the headlines at face value, I guess, is the...
That's the headline of the show, isn't it?
Well, there you go. Yes, don't move past that headline. That is it. That is the take home message.